Research for student groups on bacteria. Question number 7. What is Bacillus thuringiensis? How is it related to other species of bacteria? Talk about the microbiology of this bacterium. Do the same for Streptomyces hygroscopicus. The picture on the left is of different shapes of bacteria. In this PowerPoint, we're talking about the bacilli and the Streptomyces. This project was done by Julia Burton and Maria Shafferano. Bacillus thuringiensis is a soil-dwelling bacteria and it's rod-shaped, and it's commonly used as a biological alternative to pesticides. The crytoxin in the bacteria can be extracted and used as a pesticide. It was first discovered in 1902 by a Japanese biologist. It was rediscovered in 1911 in Germany by Ernest Berliner. It occurs naturally in guts of caterpillars, butterflies, and moths. Also, it occurs on the dark surfaces of plants. Now, several strains of this bacteria can infect and kill insects, and it's the only microbial insecticide used widespread. But it wasn't commercially available until the 1950s. It's also very safe for the environment. It is unlike typical nerve poison insecticides. It acts by producing proteins, delta endotoxin, toxic crystal. It is a protoxin, which means that the molecule must be processed to some other form before the toxic activity is present. It reacts with the cells of the gut lining the insects on the plants. It paralyzes their digestive systems and the insects die of starvation. This process takes days. Inside the bacteria, the protoxin molecules collect together and form a crystal. Crystals are visible as two pyramids under a light microscope. It has endospores as well. Endospores allows the organism to survive in inhospitable conditions in a dormant state. The protoxin crystal is very strong and does not dissolve easily. However, in the gut of insects, where the pH is basic, they dissolve into the solution. When this happens, the insect's enzymes split the molecule and the delta endotoxin confers the lethal effect to the insect. These are green spores inside the bacterial cells. Now, as we said before, this bacteria affects the digestive system of the insect. The delta endotoxin binds to the epithelial cells lining the gut wall of the insect. This creates holes in the cells, and that destroys the function of the gut, creating massive cell death. The pH is modified to a more neutral level that is hospitable for the bacteria to grow. The larva is unable to eat, thus it kills the insect. The relationship with other bacteria species includes unifying characteristics, such as gram-positive. It forms endospores. It grows in the presence of oxygen, and its trivial name is aerobic spore formers. The ubiquity and diversity of these bacteria in nature, as well as the unusual resistance of their endospores to chemical and physical agents, and the production of antibiotics and other characteristics, have attracted interest in these bacteria. The genus Bacillus remained intact until it was split into several families and genera of endospore-forming bacteria. The ssRNA analysis was used. The Streptomyces hygroscopicus is aerobic and a rod-shaped bacteria. It shares many characteristics with fungi. It usually grows as chains of cells and form a network of filaments in soil and decaying vegetation. Its spores give off an earthy odor, also known as the scent of dirt. This is a picture of the bacteria. Now, some streptomyces produce antibiotics, such as streptomycin. This bacteria produces an antibiotic called mivilicin, and is used as an insecticide and to control certain parasitic infections in animals. This is also a biofilm-forming bacteria. A biofilm-forming bacteria consists of accumulations of bacteria in aqueous environments. They form when bacteria secrete slimy materials that help itself attach to moist surfaces. Biofilm microorganisms carry out a variety of detrimental or beneficial reactions under certain environmental conditions. Now, its relationship with other bacteria species. There are over 500 species of Streptomyces bacteria. They're all gram-positive, and the genomes have a high GC content. Found mostly in soil and decaying vegetation, and most of them produce spores, and they're characterized by a complex secondary metabolism. They produce over 2/3 of the clinically useful antibiotics, and they're very important decomposers. This is a picture of the bacteria. That's it for today.